I think that the whole field, sort of immunotherapy, right, and thinking about harnessing what we already have from the immune system, for a long time was actually thought to be not possible, right? People kind of thought, well, if your immune system could actually, prevent all these different things, why didn't it evolve that way naturally? And there was just a lot of misunderstanding about how to leverage the power of the immune system to be able to fight disease. And I think over the past, I'd say about 30 years, there's been kind of, just a very rapid and strong evolution in the mindset about, how the immune system can really be leveraged against various diseases. But also there's been just revolutions in the technologies and tools that we have to interrogate and study and understand the immune system. And really it's that understanding, particularly at the molecular level, that my lab is interested in, that's empowered us to be able to now manipulate the immune system in ways that could be useful in different therapeutic applications. One exciting application is we've designed a cytokine antibody fusion protein, which is a molecule that literally puts together a cytokine and an antibody. And this molecule actually specifically activates the immunosuppressive arm of your immune system. So essentially, it expands the number of cells that you have in your body that actually prevent the immune system from going into overdrive and destroying your own healthy tissue and organs. And we've used this molecule in a number of different models in type 1 diabetes, in ulcerative colitis, and more recently in graft versus host disease, so for transplantation applications. And we're very excited about how it's been able to prevent the immune system from being overactive and to mitigate the severity of disease in a number of different models. So we're moving that molecule forward. We have work going on in glycoengineering, where we basically use glycosylation as a tool to manipulate the way that antibodies work and to actually increase their functionality. So increase the amount of time that they'll spend in the blood so that they can have activity over longer periods of time. And also to increase their targeted specificity towards certain cell types to be able to destroy, for example, cancer or chronic infections or other pathogenic agents that can show up in your body. Really, every disease in some way, shape, or form interacts with the immune system and involves things at the molecular level, right? Protein-protein interactions, protein signaling pathways are essential to all sorts of health and disease applications. And so really, I see this going in so many different directions, not just in our lab, but in other labs, as well as through industry, collaborations and interactions, being able to really address a lot of important questions that we haven't had the tools or the resources to really look at before with some of the new technologies we're developing.